Hi all, we're on to Chapter 3. The focus of Chapter 3 is cells. How did we get here? Well, going back to Chapter 1, we saw that we can consider biology at different levels of organization. Last week, we focused on the most fundamental of levels, atoms and molecules. Let's do a quick review. What is common to all atoms of an element? What are the conditions for atoms to be stable? What are the four types of bonds that we discussed? What are the characteristics of each bond? That is, how do electrons come and go? Which type of bond has charge? Which type of bond is weakest? And what are the four classes of biological macromolecules and their characteristics? I'll let you go back to Chapter 2 and review these topics. This should be an indication to you that the strategy of cramming for a quiz and then forgetting the stuff as soon as you leave the classroom door, that won't work very well in a biology class. The things that you learn in 1 week will be important in understanding the information that you learn the next week. And for those of you going on to take microbiology and anatomy and physiology, the learning that you accumulate throughout our class will be important in hitting the ground running in those classes. And then, when you get into nursing or other healthcare programs, those first quarter instructors will assume you know your biology. Well, this week, we'll focus on the next levels of organization, cells and their structures. And in lab, we'll focus on cells and tissues. Cells were discovered more than 350 years ago. Robert Hooke coined the term cell to describe his observations of cork cells. Hooke's drawings of cork cells is shown on the left, and one of his microscopes is shown in the lower left corner. Cork is part of plant stems that are made of dead cells. The first person to document living cells was Antony van Leeuwenhoek. He documented many types of microorganisms. The image on the right is one page of van Leeuwenhoek's drawings of bacteria, and a replica of one of his microscopes is in the upper right-hand corner. Van Leeuwenhoek recognized that these invisible beings must be alive. He didn't actually observe them just sitting still. He saw them live as active organisms. He saw big microorganisms like paramecium, the big cells in this view. We now call these large cells that have nuclei and other internal structures like the contractile vacuole, we call them eukaryotic cells. Van Leeuwenhoek also saw smaller things that were obviously also alive. These are now called prokaryotic cells, like the bacteria shown in this view. This video is similar to what Van Leeuwenhoek must have seen. By the 1830s, when more people had more sophisticated microscopes, it was apparent that many living things, including humans, seemed to be made of small microscopic parts. That led many scientists to suspect that cells were important. Schleiden and Schwann independently formalized this thought into what is now known as cell theory, the concept that all living things are made of cells. We now consider cells the fundamental units of life. Microscope technology has advanced a lot since the time of Hooke and van Leeuwenhoek. We now have compound light microscopes that take advantage of magnifying powers of multiple lenses. We'll use these in lab to view things up to 1,000 times bigger than real life. We'll discuss the parts and the proper use of compound microscopes in lab. Another type of light microscope is the dissecting scope, shown in the middle. These don't magnify as much, up to about 50 times bigger than real life. But they do allow the specimens to be moved and worked upon while being viewed under the scope. And yet another type of microscope, developed in the 1930s and improved upon since then, is the electron microscope. These use electron beams rather than visible light to illuminate the specimen. This enables magnification up to 30,000 times for scanning electron microscopes and even up to 10 million times for transmission electron microscopes. This study of cells isn't just academic or theoretical. The main photo in this view is of normal cervical cells and of cervical cells infected with human papillomavirus. These microscopic views are what doctors look for in a pap smear when they're testing a woman for possible cervical cancer. Understanding disorders, like cancers, requires an understanding of cell structure and process. That is the foundation for the whole rest of this term. And we'll move ahead with that in the next video.